So we're going to get things underway here. The first order of business is we do have a couple of openings for the lunch. I've already heard a couple of, I know, so if there's people that are interested, you can check with me afterwards. We'll do a little head count on that, but and that's over in Upson 331, I think, with northeast corner of Upson on the third floor. So to begin with, it's really nice to be able to bring people back who've had some sort of direct connection with Cornell, and our speaker today, Professor Deborah Sills from Bucknell, is one of those. And so I bet most of you have not had the kind of experience she had in growing up. How many of you know where Montana State University is, first of all? Yeah, all these old-timers here in the front row. But so that is one of the state institutions of Montana in Bozeman, very interesting place to go to school. And like Cornell, if you took University of Montana and Missoula and put it with Montana State, you'd have Cornell. Okay, so, and they're separated by quite a few hundred miles, to say the least. So after that, she started out in New York City, but she also went to Israel and spent a fair amount of time there, was in a kibbutz for a while, but also served in the Israeli Air Force. So if you want to ask those kind of questions, you can cover those too. Then came to Cornell and worked in civil and environmental, did her PhD thesis in working with Jim Gossett over in who was interested in feedstock logistics, anaerobic digestion, and those kind of sort of biochemical, biological conversions. And then we were fortunate enough to talk Deborah into coming over and working in our group. About that time, there was a great interest in algae as a feedstock and also as sort of something that was going to use the same kind of land for biomass. And so Deborah got very interested in that. She sort of led a pioneering effort we made in the life cycle area of this, and was one of the first people to introduce uncertainty into how you deal with a multi-step, multi-chain, sort of energy chain analysis of life cycle. Algae is a kind of biomass for sure, but it's very different. Has a lot of challenges all the way along the way. And then following that, we talked her into moving into the thermochemical side a little more directly and got involved with some experimental work in our group. Stayed on as a postdoc, and then eventually took this opportunity to depart for Bucknell and became a junior faculty member there. And from what I hear, she's doing quite well and very popular. Bucknell's a different place than Cornell, but it has a strong reputation in undergraduate engineering, and so she's in the civil and environmental engineering department there. So we're fortunate today that she's going to give us a look at sort of at the food, water, energy nexus and how humans, as well as some of our animals, have had an impact and how we might improve that. Cornell, as you know, has a lot of people. We have 30,000 of us here if you count everybody. And we have a lot of cows, too. And there's a lot of cows in New York State. Cornell's responsible for 600 of them. And there's quite a few more than that in the state of New York. I think over a couple of 100,000 or so in our near vicinity here of dairy cows and more beef cows. So they all have some challenges in terms of sustainability and energy recovery and nutrient recovery. So thank you, Deborah.
Okay. Thank you, Jeff, for introducing me, and thank you all for coming here during the lunch hour. So I'm going to talk about sustainable bioenergy, and I'm going to start with an introduction and some motivation. So this is from the Paris Climate Talks, that sustainable deployment of bioenergy can enhance energy security and reduce climate change impacts. And so just to make sure we're all on the same page, I want to give you just a definition of what bioenergy is. So bioenergy is energy that comes from biomass. It can be derived simply by combusting biomass, burning wood in your home. That would be bioenergy. Or what I'm going to talk about today is converting that biomass into a gaseous or liquid fuel and then combusting that. And biomass is just any kind of organic, decomposable material. Like Jeff mentioned, I still work on algae, but today I'm going to be talking about waste biomass. And I have up here images of 3 kinds of waste biomasses: food waste, which I won't talk about today, but it's very important, and Jeff's group is working on that. Human waste, which is hard to see this picture. Maybe when it's bigger you can see it, but it's two children doing their thing in the streets of Mumbai. And animal waste. And still a little more motivation. I have here the UN Sustainable Development Goals, and I've passed out some of them, but for those of you that didn't get one, you can look at them up here. And I'd like you just to do a brief thought experiment, talk to a friend. I'm going to give you really 30 seconds, and try and pick the top 3 most important of these Sustainable Development Goals. Just go.
It's all been
All right, anybody want to share what they think is the most important one? Sure.
Gender equality and, well, actually, gender equality and quality education are like, should be like, those should be put together.
Gender equality, quality of education. I'm not going to argue with you. Anyone else have something different? Yes.
Water and sanitation.
Water and sanitation. Yeah, I like that one. I like the gender equality one too. Yep, and there was someone over here.
Oh, I was thinking sustainable cities and communities.
Yeah, so it was kind of a trick question. These are all important. There's no most important one, but I think it's good to think about it. So the work I'm going to present today mostly focuses on the water and sanitation, specifically the sanitation as well as energy. But when I was thinking about it, because I'm going to talk about nutrients, things like fertilizer, and Jeff mentioned this, the food, energy, water nexus, it spans out too. Okay, so my talk today, I'm going to talk about 2 kinds of waste biomass, I already told you that, and 2 resource recovery processes. So the first one is a study that I did at Bucknell with Bucknell students, and we don't have PhD students, so I have quite a number of undergraduates and 1 master's student working with me on this. And we recovered energy from real domestic wastewater using anaerobic processing. So these are microorganisms that live in the absence of oxygen and convert organic material into energy. And there's some challenges when it's wastewater versus sludge, which is more often used with anaerobic digestion. The second process is a collaborative process that I've worked on with Jeff Tester's lab, and this is recovery of energy and nutrients from animal manure using a thermochemical process called hydrothermal liquefaction. And the data I'm going to show you is data that my undergraduate student collected with the help of Jeff's postdoc and graduate student, but mostly focusing on the nutrients. I'll talk a little bit about the energy, but I'm really going to focus on nitrogen today. Okay, so let's start with the first topic. So before I start, I said I have a lot of students, and that is Al Gorn. He is not one of my students, but we were at a climate march in New York City, and we saw him there. But I just want to thank the students that worked with me on this project. I'm not going to name them all, but I did not do this alone. So some motivation for this specific project. Well, there's a lot of people in the world who don't have access to a toilet, and this is very problematic. It's been, in fact, linked to malnutrition. Open defecation, that is, linked to malnutrition. Even children that have enough food to eat, there's research that shows their growth may be stunted, and the hypothesis is that they're exposed to all sorts of nasty pathogens throughout their life. And in addition, and related specifically to my work, 80% of the wastewater that's collected is not treated in the world. So this is people who have toilets, pipes, and it's collected. It's just released to the environment, and this causes environmental problems and human health problems. And so we wanted to look at life cycle impacts for 2 low-cost technologies, low-energy or even energy-producing technologies. One is an anaerobic process. It's called an anaerobic baffled reactor. This is a picture in South Africa. It's underground. This man is standing on the lid, or near the lid of the reactor. And the second is an aerobic process. Both of these are biological processes. processes that uses oxygen, but is lower cost than what we typically use in the United States. And this is one example of a fixed film process called a rotating biological contactor. This is from a clinic, a Partners in Health clinic in Haiti. So in addition to experimental work that I'm going to show you, we wanted to look at the life cycle impacts. And so we did what's called a Lifecycle Assessment and a techno-economic analysis. And for those of you that are not familiar with this, when we do a life cycle assessment and a TEA, or for now I'm going to say LCA and TEA, we start by looking at all the material and energy inputs and outputs for the process over its lifetime. And this can actually be done for a process, which I'm doing in this case, but also for a product. You might want to, do a life cycle assessment of your laptop and compare it to another laptop. And that's key. I refer to it as a semi-quantitative method because it's better for comparing two processes than it is for saying exactly, your laptop will emit X grams of CO2 equivalents over its lifetime. It's better to say, the Dell laptop is better than the Apple laptop by this much. Now, when we talk about the life cycle, a product or a process has a life cycle like we do. So our life cycle is we're conceived, we're born, we're go to school, you get a job. Some of you might be thinking about jobs. Hopefully we retire, and I want to live in the Caribbean, and then eventually we all die. So that's just what happens. So similarly, a product or process starts with extraction of raw materials from the earth, manufacturing stage, a use stage, if it's something like a laptop, perhaps a transportation stage in there, and then an end of life or decommissioning of a plant. And so we want to analyze, again, all the inputs and outputs, material and energy, over this life cycle. And for the cost, we'll then do an engineering cost analysis. And for the environmental impacts, we will convert all of those energy and material flows into environmental impacts. So the anaerobic baffled reactor. This is, there's a national lab in Switzerland called EVAG, and this is one of their recommendations for a unit process that can be implemented. It's been used so far pretty widely in South Africa. There's also a nice case study in Nepal. What it does is it converts the organic material that's in the wastewater into methane gas. Now, some of you may be familiar with anaerobic digestion. It's exactly the same process, but it has different problems that anaerobic digestion does not have because this organic waste is very dilute. It's much more dilute than a digester. And what happens is that some of the methane, or a significant fraction of the methane, gets trapped in the water and is not released and collected as gas. And methane is a very potent greenhouse gas. Per mass basis, about 30 times more potent than CO2 when it's integrated over 100 years. And so having methane left in your wastewater, and then when it's discharged to the river, then it will come out into the atmosphere. So it could represent a climate liability. So we wanted to look at this with life cycle assessment. So we have this really nice ABR. It's about 20 liters in the lab. We were thinking about, this 80% of wastewater that's not treated. We were also thinking about low temperatures. One of the reasons we were thinking about lower temperatures, 15 to 20 degrees C, because we know there's additional challenges with anaerobic reactions at lower temperatures. And there hadn't been much published on this, so it seemed like a more novel research project. And so we wanted to see, can it remove the organic material and clean up the water reliable, and also can it convert efficiently or a good amount of that organic material into methane, which could then be used for heating, for cooking, if you're a large plant, even for production of electricity. Now, we wanted to compare it to something, and we thought, okay, we'll do a fixed film process. I showed you before a picture of a rotating biological contactor, which was really what I wanted to use, but we had data for a trickling filter. In fact, it's in Ithaca. It's not the main wastewater treatment plant, but it's at Bolton Point. This is a picture from there. So we compared the treatment of this domestic wastewater for the model. We used our lab data for the ABR, and we used the plant data for the trickling filter. And so here's just a schematic of our ABR. The most interesting part of this schematic is what you can't see is my students driving to the wastewater treatment plant every day and bringing wastewater. Because a colleague of mine, yeah, that was fun, especially when it's spilled in the hallway. The custodians do not like me at Bucknell, I'll tell you that much. So a colleague of mine had already done studies on it with synthetic wastewater, but we wanted to see when wastewater concentrations vary widely, and they do, because we have combined sewers where I live. So when there's a rain event, the wastewater might be very, very dilute. And when the wastewater plant receives trucked waste, because it does, there's like really crazy spikes. So I wanted to see, can it be robust with varying waste? All right, so here's some data. We ran it for about 100 days where we thought it was in steady state. So on the x-axis, we have the days. On the left y-axis, we have the effluent COD. COD is just the organic material in units of how much oxygen it consumes. Any environmental engineering people here? Okay, so you've heard of COD. And I know there's one other who has. But just think of it as organic material. So the black is the influent concentration in the same units as the blue scale here. And you can see this wide variation that I was talking about. But the blue looks pretty steady. We removed between 70 and 80% of the COD. This is the percent removal, so this y-axis shows the percent removal. There was variation, but it worked. The sad news is that for the United States, the regulation is 30 milligrams per liter of organic matter in units of COD, and we were more at 120. So this is not sufficient. But we argued in the paper that, oh, this is better than nothing. Are these too stringent? But we do think this needs a polishing step. But the reason it needs a polishing step is really in this pie chart. So this turquoise and yellow represents the fraction of organic matter in the influent. The whole pie is all the organic matter in the influent that was converted to methane. So the good news is that about half of the organic matter was converted to methane, and that is what we expected. It's a little more than half of it. The bad news, though, is at this cooler temperature, about 1/3 of the methane was trapped in the dissolved phase. And so that represents a loss of energy. But what you'll see in the life cycle analysis, it really represents a climate change liability, so that's not good. So then we did our life cycle study, and we looked at 4 assemblies, I'm going to call them. One is just the anaerobic baffled reactor on its own. One is just this representative fixed film process on its own. It needs some other, it needs a primary clarifier, but more importantly, aerobic processes themselves produce biomass. So in a sense, they take the soluble waste in wastewater and convert it into microbes. And then we can take those microbes and do anaerobic digestion to recover energy. Then, and this was the one I was most interested in and still am, was the anaerobic baffled reactor, followed by the fixed film process as a polishing step, both to oxidize that methane and to remove that organic matter, because we had 120 milligrams per liter and we'd like to go to 30, which is the regulation. We also looked at a constructed wetland because we saw in Nepal they had an ABR and a constructed wetland. The constructed wetland, however, would not address the methane removal. It would only remove more soluble organic matter. That's not methane. So in life cycle assessment, you have to choose what's called a functional unit, and it's simply the parameter and the value of that parameter to which all your flows are normalized. So since we modeled a 2 MGD plant, which is not a large plant, and the reason we did 2 MGD is because people told us, oh, you shouldn't do anaerobic digestion if it's slower than 5. But we had seen some people that were doing it, and we wanted to show that the economics might work for that. We did not model removal of nitrogen and phosphorus, which is something that needs to be done here in the United States often. We used a database called EcoInvent to get impacts for things like electricity, cement, polymer. I'm calling those background or maybe upstream processes, not things that directly happened in our plant. The electricity, in some cases at least, was not produced on the plant. For our actual process data, we used our lab data from the anaerobic baffled reactor that I showed you, and we had some modeling equations plus plant data that we used for the fixed film process. So then you need in Lifecycle Assessment what's called an impact assessment method that will convert the flows into an environmental impact. So we use the IPCC 100-year method to convert all the emissions into a climate change impact. And we used a method called Impact 2002 Plus to look at 3 other impacts, which were energy depletion, primary energy depletion or production, human health impacts, and ecosystem impacts. And we did uncertainty analysis because, Jeff taught me I had to do that with my algae work, and so I've taken that to heart. Okay, so here, this is just a schematic of what went into the ABR, what went into the fixed film process. I'm not showing you a schematic for the constructed wetland, but it was pretty simple. The yellow are the inputs, the pink are the inputs, the yellow are the outputs, and the blue are the processes. How do we do uncertainty analysis? Well, all we did was we input into our model, we just did parameter uncertainty. So we input our parameters as probability distribution functions. When we didn't know a lot, we input them as uniform or triangular distributions. When we had more data, we did normal and log normal distributions. And so if you imagine I have a model that's 1 plus 2 plus 3, or maybe times 3, each time I run the model goes and randomly picks a variable for parameter 1, then randomly picks a variable from parameter 2, then for the third parameter, we had many parameters, calculates the model, reports a result. And you do that 1,000 times, and then you get a distribution. I'll show the distributions as these box plots because they're more condensed. They're not a traditional box plot. They just represent the 5th, 25th, 50th, 75th, and 95th percentiles of the distribution graphically. All right, so let's look at, I'm just going to show you detailed data for the energy and the climate change. So on the x-axis, we have the trickling filter, the fixed film process, the anaerobic baffled reactor, the anaerobic baffled reactor with the trickling filter as polishing, and the anaerobic baffled reactor with the constructed wetland as polishing. And life cycle assessment, you kind of have to turn your head upside down. Negative is good, and positive is bad. I have one colleague who's always asking me to change this when we report stuff, but that's the convention. So as an LCA person, I tend to leave it. So for the ABR, we have this energy production. We modeled a CHP system, and we produced electricity, and we gave it the impacts of the electricity grid. I'm not sure which data this is. We did it for Pennsylvania and for India. The magnitudes look, the pattern is the same, but let's say it's for Pennsylvania. So we have this benefit. We also have a benefit from the anaerobic digestion for the trickling filter processes. But the trickling filter uses more energy than the anaerobic baffled reactor does. It's a very simple process. It's an overall energy producing processes. But actually, all of these are overall energy producing. The values with negatives exceed the values with the positives. But now comes the sad and interesting part. So this is all overwhelmed for the ABR, the anaerobic baffled reactor, and the anaerobic baffled reactor with a constructed wetland. But let's just look here with this huge impact for the climate change impact due to that dissolved methane. Now what we assumed for our trickling filter model, we assumed it was magic. Well, magic. It was amazing, not magic. It was really good, and it removed all of the methane. For the constructed wetland, we assumed that it didn't remove any of the methane. And we see that to make the ABR work, we do need to remove the methane. Ideally, we would want to recover that methane, but because people argued with me that a fixed film process would not remove the methane because it would all volatilize, I stuck with this. And I stuck with this, and we're now doing experiments with a rotating biological contactor. First time I showed this to a environmental engineering professor from Michigan, she said to me, yeah, an RBC has a better chance of working. You can ask me why than a trickling filter, but I'll leave it at that. So basically, this is the climate change impact. Sorry, I forgot to say that. It's bad with the ABR. Then we did uncertainty, and you really can't, so now I switched, instead of up and down, I'm switching horizontal, so now you have to turn, I don't know, in your mind this way. But I have climate change, resource. These are the impacts I showed you. I'm also showing ecosystems and human health. I will tell you that human health impacts are more uncertain. The methods where you convert flows into impacts is more uncertain than climate change and resources. For something to be different, it has to be 3 orders of magnitude different for me to be able to tell you, oh, this one's better for human health than that one. And the ecosystem, and for climate change and resources, it's really only 10% different. And for resources, I think it's, for ecosystem, somewhere in between. So we can't really see anything here except what I showed you before, that for climate change, it's so, the ABR is so bad. Oh, the other thing we did here is we normalized all of these impacts before they were in their real units, I don't know, megajoules or kilograms of CO2. Now I normalize them into what's called LCA points. And all that represents is an estimated impact of a human being in the United States over a year. And it's just so we can look at all the impacts together. There is a judgment on this. This isn't an absolute value, but it's a way to compare things. So all we can see is when we look at that, these impacts from the methane from the ABR far outweigh any other impacts. And it's not very interesting. I can't tell what's going on there. So if I take that away, I can still say, wow, I have a lot of uncertainty. The trickling filter on its own, however, maybe is worse. That's the top one. We have trickling filter, ABR, ABR plus trickling filter, ABR plus constructed wetland, LCA points. The trickling filter maybe is a little worse than everything else, but really uncertain.
Question, is this a log scale?
Nope. These are just these points. They're normalized. There's like, I don't know, I don't remember the number, but like 10,000 grams of CO2 equivalents in one LCA point.
Thank you.
Yep.
What was the vertical red line?
Oh, yeah. Thank you. I should have said that. The vertical red line is the 0. And if we look here, too, and it's the same thing what I said, positive is bad and negative is good. So anything that's below 0 is good. So overall, this looks good. That's a very important question. Thanks. But this climate change from the methane is so bad. Yep. Yeah.
You said you took the parameter from the parameter distribution.
Yes.
How do you get the distribution for those parameters?
So for the, let's say, methane production per COD removed, we have our data. For other things, we look in the literature, and we might just use a uniform distribution if we don't have much data. You know, let's say, oh, mixing takes, I don't know, 8 kilowatts for the reactor, and I find another paper that says 2 kilowatts. You know, I'll just do a uniform distribution, so equal probability straight line. If I find more data, I might just do a triangular distribution. So this would be most likely something like that. Okay? Yep. Yeah, takes a lot of time. Okay, we also did economics. And the economics here, the net present value of each of these systems, which is again ABR, trickling filter, ABR plus trickling filter, ABR plus constructed wetland, that's on the x-axis. On the y-axis, we have the sum of all the LCA points. So we gave each one of them equal weights. Someone might not like that, but just for a thought experiment. And so what we see is that, as expected, the ABR and the ABR, the constructed wetland, are the least costly. They're the most to the left here. The ABR and the trickling filter are more expensive. And then with the impacts, the ABR plus trickling filter, because we got rid of that dissolved methane, is the best from an environmental perspective. Okay, so our conclusion from this is that if we want to develop sanitation, you know, so we don't have children with stunted growth and don't want them exposed to all these pathogens, and we want to clean up wastewater, we also want to think, we don't want to turn that into a climate change liability. So this is horrible for people, what we see here on the left, but what we see on the right isn't great for people either. And that's one thing I really like about LCA, because it's good for identifying processes that don't cause burden shifting. So I don't fix one problem, but turn it into a different kind of problem. Also, if we want to work with the ABR, and we love our little ABR in the lab, we've got to do a post-treatment.
There's lots of options for that too, right? Other than you could thermally heat it if you use some of the
Sure, yes.
biogas and get the
Yeah.
methane off gas and use it that way. But
Yeah, yes.
But it's another cost, but
Yeah, yeah, that is an option that would be good to look at.
but not so simple for this kind of system, right?
Yeah, but this may be not so simple too, but this I was curious about because I had an argument with someone whether it would work. So now we have a rotating biological contactor in the lab. We want to see if we can oxidize that methane. Although in the picture here, and typically they're open, so the bugs, the microbes get exposed to oxygen. We had to close ours and pump air in there because we're trying to do a mass balance on methane. Although this is what it looks like when it's open. This is all the microbial growth on it. This is just from a couple days ago. So we're measuring the oxidation both of that remaining 120 milligrams per liter of organic matter and the methane. So this is the organic matter. So we came in, we simulated, now we're not using real wastewater, we're simulating the effluent from our ABR because our ABR isn't running these days. It was a lot of work to have people pick up wastewater every day. And we're getting 95% of the organic matter removed, which I was not surprised at all. This is not that interesting. We expected this to happen. It's also below the 30 milligram per liter regulation. But this, it looks like we're getting 80% of the dissolved methane oxidized. Our control isn't perfect yet. We're rerunning our control now to make sure. So this is really just recent, very, very recent data, not published data. So yeah, so that's it with my wastewater project. Sorry, that's missing. We're updating also the life cycle assessment with actual data for rotating biological contactors since we used a different fixed film process for the published study that we did earlier. All right, I go till 1:10, right?
20 minutes.
Yeah, all right, so we're going on to, I can take one question if someone wants to ask a question before I go on.
Describe the fixed film
Yes.
process for just a second,
Yeah.
how it works.
Yeah, so these discs, they're about 40% submerged in the wastewater as it flows through. So they rotate, and as they come up, they get exposed to air, to the oxygen they need, and then they go back in and they treat the wastewater. So it's a very slow rotation.
A minute?
It's like an RPM, I think, 1 to 5 RPM. I know my motor is, but yeah. All right. All right. We can do more questions at the end.
And they can come to lunch too if they're.
And you can come to lunch too. There's pizza here. All right. So this is the project that I've been working on with Jeff Tester's group, recovery of energy. And the analyses we've done at Bucknell have really focused on the nutrients from animal manure using hydrothermal liquefaction. So I want to thank up front Jeff Tester. Without him, none of this would have happened. I really enjoy collaborating with his group. And Dr. Roy Postmanek led the running of the reactor work. And after Roy left, Borja Cantero, who's a PhD student, Roy's in Israel now. He's a PI at a national lab in Israel, and we're still collaborating. It's a little further away, though. Borja ran reactions for us after Roy left for this data that my students and I are now analyzing at Bucknell. All right, so we have a lot of manure in the United States. We have a lot of animals. This is problematic. We've really seen this recently with Hurricane Florence, with flooding and manure getting everywhere. This causes environmental problems like eutrophication. Here's a recent article about an algal bloom in Owasco Lake, not too far from here. This is unpleasant. This makes you not want to go swimming. This also leads to problem in drinking water. There's toxins that algae produce called microcystins that are really tricky to remove from drinking water. So not good, all this manure. We collaborated with Brian Schwartz from Johns Hopkins Public Health School, and we collected data. This is a bad-looking map of central Pennsylvania served by the Geisinger Health System. And we collected data on manure, where it's produced, where it's applied, and it was linked to human health problems because my collaborators from Geisinger and Hopkins have the health data. So one study linked poultry manure to pneumonia, community, I think it's called community something ammonia. The other study linked it to Campylobacter-derived diarrhea. And the third study linked dairy and swine manure to soft skin tissue infections.
This is not a third world country.
This is, well, it's Pennsylvania.
Right.
I don't know. We can make Pennsylvania jokes. So yeah, this was not very exciting work for my students, but it was good for first-year research students. They actually drove around to county conservation districts because this data is not digitized, and scanned the data and put it into Excel. And yeah, so we have very estimated local data. Okay, but how can we turn this horrible problem into something exciting, right? Let's recover energy from it. Let's maybe recover these nutrients that are causing these algal blooms. So we can do this. So we already, in a lot of farms, and there's people in this room working on this, how much we do, we convert it into biogas using anaerobic digestion. Typically, however, anaerobic digestion digests or converts about half of the organic material that's in manure into energy. And so we'd like to valorize or take advantage or utilize or use, whatever your favorite word is, more of it. And we're looking at converting it into, or we've been looking at converting it into bio-oil with a process called hydrothermal liquefaction. There's been a collaboration between Cornell and University of California, Riverside, taking what's left over after you produce the oil and doing some membrane distillation to get out nutrients and clean water and really close the loop and have a circular system with manure. So that would be awesome. So hydrothermal liquefaction takes advantage of the interesting properties of water in the subcritical region, where water acts more like a polar solvent and can convert the carbohydrates and the proteins in animal manure into a bio-oil. And this bio-oil is different from crude oil. I would say it's similar to crude petroleum oil in that it has to be refined and upgraded so it can be used in our cars. And so we take our biomass. These are components of biomass. We have this in Snee Hall, this nifty HTL reactor, and it produces 4 phases. We have not been analyzing the gas phase, but we've been analyzing the aqueous phase, the crude oil phase, and the char phase. So Dr. Roy Postmanek did a number of papers on this. And here's some results from one paper we did. And he looked at running the HTL without a catalyst, or with an acid catalyst, or with a base catalyst. And what he saw that was for the acid catalyst, so these are my 3 conditions: no catalyst, acid catalyst, base catalyst. On the y-axis, I have the carbon recovery. We would like most of our carbon to be in the oil phase, because the carbon, you could think of it as the energy that can be combusted. And with the acid catalyst, we got more of the carbon in the oil phase. Based on some of our analysis of the aqueous phase, we think this had to do with the production of a chemical called 5-hydroxymethylfurfural. We're trying to understand this better. But basically, it seemed like the acid catalyzed had some advantages. And that was the motivation for this study that we did, I'm going to show you data for, that we were looking at the effect of the retention time on the hydrothermal liquefaction of manure digestate without a catalyst and just with the acid catalyst. The base catalyst didn't help much. So we want to look at the nitrogen and carbon distributions in the oil phase and the aqueous phase. I'm going to really show you data for the aqueous phase today. We're having some problem with our oil samples. And what we'd really like to know more about is, how does this happen? So we know hydroliquefaction happens, but we'd like to understand the mechanism. So we thought if we took multiple time points, like 5 minutes, 10 minutes, 15 minutes, and we see changes, maybe we can try and begin or better understand what's happening. So here's our experimental setup. We have this manure digestate. I never said, but digestate is what's left over in that anaerobic digester. We ran the HTL reactions here at Snee Hall. Borja led this under the supervision from far away of Roy at 5, 10, 20, 30, and 40 minutes. We separated the oil and the aqueous phases. Here we didn't have a solid phase because we were just taking time samples straight out of the reactor with a heat exchanger. And then at Bucknell, we are analyzing the oil phase with GCMS. I won't show you any of that data. And we're analyzing the aqueous phase for just carbon, nitrogen, phosphorus. And we're doing HPLC and LCMS to, again, tease out these mechanisms. And the data I'm going to show you today, my student, I will name her because she's done so much of this work and she's so awesome. And hopefully she'll apply to Cornell when she She wants to get a PhD. She already knows she does. She's not sure where she's going to go. But we're just going to look at the carbon and nitrogen in the aqueous phase. And what's really interesting here from the sort of tester group perspective is the nitrogen. We haven't looked at that before. So here's nitrogen with no catalyst and with acid catalyst. So on the x-axis, I have the different retention times. And on the y-axis, I have the percent nitrogen recovered. What I see is for both with and without catalyst, as the reaction goes on, I get less nitrogen in the aqueous phase. We're not sure yet, but we think that means more nitrogen in the oil phase. More nitrogen in the oil phase is bad, because that's something that has to be removed in the upgrading process. You don't want nitrogen in your fuel. More nitrogen in the aqueous phase is good, because then we could perhaps recover it as a fertilizer or use it as a fertilizer. We'd probably take it out of there somehow first. With the acid catalyst, it looks like we have more nitrogen most of the times. Also, we measured what form the nitrogen is in. Organic nitrogen, which is the top pink bars, is not very useful to us because it can't be taken up by plants, but ammonia and nitrate can. So the two bottom purples, we're happy that most of our nitrogen is in the mineralized and useful form. So that's good. We also did this for carbon. These are on the same scale, so right away you can see we're not recovering as much carbon in the aqueous phase. That's good news, because we want that carbon to be in the oil phase. Maybe with the acid we're getting a little bit less. But what's really interesting here is the trade-off. So I want more nitrogen in my aqueous phase. So you might say, oh, do a reaction at 5 minutes. You maximize your nitrogen recovery. But I want as little amount carbon in the aqueous phase. So then you look at this and you say, do 40 minutes. So maybe an LCA and TA model can help us, make some decisions. Or maybe we just have to think and talk about it some more. So this better understanding of the reaction mechanisms, we've just started this. These are just some initial HPLC data. They're not all that interesting. What they are interesting is that on the x-axis, we have just concentrations. On the y-axis, we have the reaction times. We are seeing different types of compounds with the catalyst and without the catalyst. We expected that, but we see more organic acids when we have the acid and more monosaccharides with no catalyst. And yeah, just the really tip of the iceberg. So, in conclusion, we can recover energy and nutrients and turn this horrible problem into something good. The costs are challenging. I'm not showing you cost analyses. We have done some work on that. I think Roy's presented some of that in other versions of this seminar. So it's exciting, and we have a lot more work to do, which is good, because I like collaborating with Jeff. So, oh yeah, and then we have lots of time for questions.
We have plenty of time. And we have a room full of people that could take some interest in
Any
this.
catalyst available to you other than the acid?
You mean, could we use like solid catalyst? Yeah, we're talking about, he's doing that in Israel,
That
and
would
maybe we'll
present
do that
some
here.
challenges to of recovering it and things, but it's worth it. There are other groups that are looking at this too, ruthenium and.
Do I have a preference for catalyst? No, we haven't really thought much about, there's people who use like rhodium.
Yeah, ruthenium and rhodium. Ruthenium's cheaper, but there's a recovery issue, and how long does it last? This acid is a simple addition of acid or base, which is, although you pay the chemical cost, pretty inexpensive in terms of recovery, right?
How about students?
Yeah, back here.
Yeah. Wait, a unit. So the different process. So the biological process takes longer. So although ours was so dilute, the retention time of our ABR was half a day. That's pretty short for a biological process. Anaerobic digestion is usually on the order of 15, 20 days. The hydrothermal, this is what's exciting. They represent higher capital costs and more specialized equipment, but you saw it was minutes. So really what HTL is doing, that's in a way exciting. If you think about the petroleum oil that we have under the ground, it was really biomass that was compressed and heated up in the ground and converted over millions of years to fuel, and we're trying to make that happen in a matter of minutes with this type of reactor. Any questions over here? Yeah.
In terms of the anaerobic Baffle reactor, is there a way to use the methane that you produce from it that isn't trapped in the water to increase the temperature within the reactor?
Yeah, that's what Jeff suggested. That we thought about that. We didn't model that, but that's actually probably a good idea. We would still get methane trapped because the percentage of methane or the concentration of methane in your liquid stream is a constant. It's based on Henry's law, if any of you studied Henry's law. And so when you have a dilute stream, that concentration, a dilute stream of organic matter coming in, it represents a larger fraction of the total methane you're producing than if you had a whole lot of methane. So if I had 1 million tons of methane, or if I had 1 ton of methane, the concentration of methane in that liquid stream is going to be 18 milligrams per liter. So when I have 1 million tons of, let's say, potential methane, it's not much of it. It's who cares? It's a tiny bit. But when I can only make 1 ton of methane out of my wastewater, and I have 18 milligrams per liter, it could represent, and it does, it was like 1/3 of it. So yeah, but Henry's constant is a function of temperature, so heating could help quite a bit. So that would be an interesting modeling. Yeah, I'll take one here and then I'll come back there. Yeah.
Can you go into a little bit more detail on why you think the trickling filter works really, really well?
It didn't. We didn't do it. We just assumed it did. Okay. But the rotating biological contactor works, well, a rotating biological contactor is a completely mixed reactor. And so the reason the methane doesn't volatilize is because if you have organisms in there that are keeping up kinetically with degrading it, you never actually see the concentration of methane that's coming into the reactor. And because an RBC is more well mixed than a trickling filter, the trickling filter probably wouldn't work that well. It was just an assumption for kind of a thought experiment. Luckily, they let us publish it. But I think we said that, we were honest that it was an assumption. And there's a few people that have argued with me that it wouldn't work in the rotating biological contactor. But from what I learned in my bioprocesses course at Cornell, I think it will, and it looks like it's working. So, yeah. I'm going to take one over here, and then I'll come back. Yeah.
You mentioned for the HTL reactions, they're being conducted in batch, right?
Yes.
So do you foresee this to be converted into a continuous process?
Yes.
Come to our lab in a year or so. The young man in back of you, that's our next step. But if I could just put one advertising plug in here, is that so you know that in addition to these seminars, we have modules that are taught. So in the spring, there will be one on biofuels, and it will focus on sort of this coupling of AD, anaerobic, and thermochemical methods. So if you want to hear more about it, if you're here in school next year, in the spring, we'll also have probably some opportunity for speakers here in February, because there will be a workshop we're running, and there'll be our friends from Israel will be here. We'll probably have the people from Riverside. So there may be a chance for more selective seminars that you could go to too. But I have one question that maybe, and okay, let's take this one. I have one left, a short one. Yeah.
I just want to ask Jeff, if I'm going to take the environment using the HDL technology, do you suggest I also include the acidic catalyzed?
Oh, of course, yeah. I'd say you want to look at all options, but we can give you some data. But remember, it's small scale data, so there's a lot of uncertainty. So when you do LCA, you have to talk to someone like Deborah or others who are very good at embedding that into it. So there'd be a base case, and then you're going to have a huge variation of outcomes for that.
My worry is that if I include the acidic catalyzed, I will have to also include the treatment for this.
Yeah, and where the acid comes from, and the sort of energy penalties and environmental, absolutely. LCA is very detailed and tedious, but that's available. You can find that kind of information.
Yeah, an EcoInvent, and Cornell has a license to that EcoInvent database. So
you haven't said much about the solid phase, and we have other experts here. Professor Lehman, and biochar and this hydrochar, that has some value too. Maybe you want to just comment on it. So we're not just throwing it away. It does have
Yeah,
value.
well, the only reason my data for the thermochemical is because we were doing a time course and we were just pulling samples out through the heat exchanger and we weren't analyzing the solid. But the solid, could be used as an agricultural amendment. I mean, biochar has shown to have a lot of benefits. I think the jury's still out on hydrochar, but I don't. Some people talk about combusting it. People have talked about using it for resins.
Carbon sequestration,
Yeah.
things like that too.
Yeah,
So there's
so,
other
yeah,
benefits.
Yeah, and in the paper we did that Roy led, we definitely talk, we include that in the analysis. But but these specific samples that I'm looking at right now were not, that's the only, I didn't not mention the solid because it has no value or no issues, because I think today I was talking with Quay that maybe there's metal in there from the
Right,
reactor.
exactly, right.
We were talking about measuring heavy metals.
Yeah.
But yeah, so it's definitely an important thing to think about, the solid
Absolutely.
phase.
So we have a few openings for the lunch. There were a few holes, should I say, in the schedule. So I think the lunch is over in Upson on the third floor, 331 or something like that, the northeast corner. So if you want to continue to interact with Professor Sills, please come there in the next few minutes. So thank you again.